Okay, well, good evening and thank you very much for the invitation. Our subject for tonight, reading bottom upwards, is effects of homeopathy for any indication. Studied in randomized placebo-controlled trials, the trials have been evaluated in systematic reviews, including meta-analyses, and we conducted a systematic review of these meta-analyses. Now, first, to explain so that we all know the technical words, a systematic review is an overview of existing studies with a structured and criteria-based approach to search, identification, inclusion, analysis, and presentation of the studies. Now, such an analysis leads to a research synthesis, which classically consists in tables summarizing key results from each study, as we shall see. But it can also include a summary effect estimate for the studies, and then it's called a meta-analysis. So all meta-analysis will be systematic reviews, but not all systematic reviews are meta-analyses. And our systematic review is not a new meta-analysis, but looking at the systematic review on the existing ones. So we shall look at the background for our work, the methods, and particularly the results. Is it working, your system? Yes. Now, effects of homeopathic treatment have been investigated in a large number of randomized placebo-controlled trials. Now, as for today, there are about 500 of them, 500. And these have been summarized in systematic reviews. Now, for reviews on therapy effects, the usual approach is not to do like what we're looking at today. That is, the usual approach is to look at one specific intervention for one specific indication. So this homeopathic product for coughs and colds or rheumatoid arthritis or whatever, and aggregating then effects in trials which are then similar because they all look at the same intervention for the same condition. These are aggregated in a meta-analysis. But for homeopathy, there has also been the opposite approach followed, looking at all types of homeopathy for any indication, aggregating all trials, which are then not always similar, except being placebo-controlled trials, into a meta-analysis. And the reasons for doing so was stated in one such meta-analysis. Many scientists think that homeopathy violates natural laws, and thus any effect must be a placebo effect. So if you aggregate all placebo-controlled trials on efficacy of homeopathy, will homeopathy then differ from the placebo groups? And that was our first research question for our systematic review. Does homeopathic treatment have positive effects beyond placebo in these meta-analyses? And our second question was, does the first question actually make sense? In other words, do the findings support this notion of such a thing as one common effect or absence thereof across different types of homeopathic treatment and among different types of indications? Because of course it could be well the case that some types of homeopathy works for some conditions and other types don't work for some conditions. So, but the underlying sort of hypothesis that we were looking at was this: homeopathy doesn't work at all for placebo, or does it? So our eligibility criteria were meta-analysis of these randomized placebo-controlled trials, including secondary analysis thereof, published by the authors or other authors. The trials could be on humans, but not animals. This was not veterinary homeopathy. All ages, any country, any setting, any indication, disease, or symptom. But there should be therapeutic benefit measured in any clinical relevant outcome. Publications in any language. So it was very broad. And we ended up with 6 meta-analyses published over 20 years from 1997 to 27. Of these 6, 3 covered all types of homeopathy. That is Linde 97, Kucherat 2000, and Shang 2005. And there were 2 restricted to individualized or classical homeopathy, Linde 1998 and Mathy 2014, and 1 restricted to all the other non-individualized homeopathy types, the last of Mathy from 2017. Now, some people wanting to criticize our work say, well, does it bring anything new? Well, it does, because what I continue telling you has not been done before. We looked at risk of bias of these 6 meta-analyses, assessing them with a standard instrument called ROBIS. Now, 3 meta-analyses had low risk of bias, in other words, high methodological quality. So that was the first and the 2 last, as you can see, the green ones. And the other 3 ended up that the conclusion is then number 5, lowest, with a low quality or high risk of bias. So this was somewhat surprising to us, this very clear dichotomy of the quality of these works. Now we go to results of individual trials. So this is the standard approach. Well, it may be in tables in a paper, but I show you here as columns with colors of looking at results for each trials and summing up how many had this and that. So what we have here is that the green and yellow columns together show some benefit of homeopathy over placebo, which was the case for between 68% and 84% of trials. So the homeopathy groups fared better in most cases. Now, the green column segments indicate what you call a significant homeopathy effect, and that is less than 5% probability of positive results occurring by chance. This is sort of probabilistic theory in statistics. And this was found, significant positive effects. It varied from 14% of studies in Mathy 2014 till 65% in the rather small meta-analysis by Kucherat. Now, the red segments indicate a non-significant effect of placebo compared to homeopathy, which was found in 16 to 32% of trials, while a significant positive say, effect of placebo, well, the positive because the patients were doing better, was found in only 1 single trial. So, as you see, if you have had a 0, if homeopathy had no effects at all, you would have a more balanced picture between the red ones and the green and yellow ones, which is not the case. But this is the traditional systematic review approach. And in a meta-analysis, all these different results are, as I said, aggregated into 1 single effect estimate, taking into account the number of patients, the effect size, and the variability of the effect sizes of each trial. Now, our first primary outcome out of 2 was the effect estimate for all included trials in each meta-analysis. And that was available for 5 of the 6 publications. And all 5 showed a positive, significant effect of homeopathy beyond placebo. Now, in trials with lower methodological quality or conversely higher risks for bias, results may be inflated or they may be leveled down, but they may be skewed systematically in one direction compared to the true results because of various systematic errors. And that's the reason why it's common to look at results after restricting the samples of trials you put into the meta-analysis to what is often called high-quality trials. And for our second research question, we looked at that and we defined it in the protocol as effect estimates among trials of higher quality, defined as such by the authors of the meta-analysis, with maximum 1 high-quality category over per meta-analysis, so that with a clear-cut separation between higher and not higher. And this category was to be based on at least 3 specified components of methodological quality. Components can be such as, was the randomization sequence properly conducted, and were the researchers blinded as to which group is the next patient going to come into, blinding or random allocations. These things are deemed very important. Dropouts and other aspects. So these were our criteria, and for this analysis, effect estimates were available for 4 out of the 6 meta-analyses. Notably, the Shang publication didn't have it, but other authors calculated and published it later from the same data. So all 4 estimates favored homeopathy, and in 3 of them, the difference was significant. But in 1 covering non-individualized homeopathy, it was not significant. So, and that's the sort of usual cutoff measure for saying this we accept. Of course, not significant findings can still be irrelevant, and they can still represent a true effect, but the convention is to draw a line between what's usually 5%, it can also be 1%. You know that in court it's different, it's often more than 50%. So next we assessed what is called the quality of evidence for positive effects of homeopathy beyond placebo. It's also called confidence in cumulative evidence or certainty of evidence. And for that, we used the state-of-the-art grade framework, assessing 6 factors, each discussed in a paper by the grade group. And these are the one we spoke about, risk or bias of the trials, and then inconsistency or heterogeneity in the findings, and risk of so-called non-reporting bias, which I cannot go really into tonight, imprecision and indirectness, and occasions for rating of the quality of evidence. So this is rather technical, and nobody has done that before among these 6 meta-analyses. So we were the first to do this, and our assessment was performed for 3 groups of homeopathy according to the scope of the 6 meta-analyses. Now, grade has 4 categories, and meta-analyses of placebo-controlled trials will start in the highest category because it's considered the best evidence you have for clinical effects, but it can be downgraded now for specific reasons. And we concluded that for individualized homeopathy, quality of evidence was rated, we rated it as high. But for non-individualized homeopathy and for the 3 meta-analyses addressing or assessing all homeopathy types, both of these were downgraded 1 step into the second best category of moderate quality of evidence. Now, the reasons were for non-individualized homeopathy, a very small number of high-quality trials, only 3, with the benefit of homeopathy no longer significant, as we just saw. So there still may be effects, but they aren't really sort of very, very solid, or not, at least not for the top grade. And for all homeopathy types, the reason was uncertainty in the Shang meta-analysis, which had 110 trials, because subsequent researchers from it's Robert Matthey and his group, they identified later 41 additional trials which were potentially eligible for inclusion, but were not identified by the Shang researchers. And it may just be because availability on the internet of data, increased rapidly in the first and the beginning of the second decade of the 21st century. But knowing that there were 41 other apart from the 110 means that you cannot know whether these 41 could have changed results if they had been added to the original meta-analysis. Now, this is called risk of location bias. It's not publication bias, because the trials had been published, but they haven't been detected or located by the authors. Now, I believe I am going to overspend. So if you have a very clear deadline at 8, we had to be very, very fast. There was some delay with starting, but if I may continue, I just continue.
Please continue.
Okay.
Yes, no problem.
Yeah. Okay, so in a sensitivity analysis, we did the same thing after restricting our sample of meta-analysis to the 3 with low risk of bias. And this is something we hadn't thought about before because we didn't know that we were going to have this clear-cut separation. So this is what you call a post hoc meta-analysis. But it really sort of begs the question to do it. And as a result, the quality of evidence for all types of homeopathy was upgraded to high quality because the Shang meta-analysis was excluded from our evaluation. The other results were unchanged. So this is a slightly more positive, with the upgraded all types of homeopathy, but it's not sort of predefined. So, you can't put it in the abstract. Now, the second question was, as I said, is this notion of one overall effect supporting by the findings in plain language for non-statisticians? Do the trials seem to belong to the same family? If you have a porridge, do you have one egg in the middle, so to speak, or do you have multiple lumps, clusters of effects? Now, this was supported for individualized homeopathy because, strikingly, although the homeopaths treated very different conditions in their patients, their uniform approach to therapy perhaps was the reason why there was still statistical homogeneity. While for non-individualized homeopathy, and that was not the case, because there you had so-called statistical heterogeneity. So you have this porridge with many lumps, many clusters in it. And accordingly, effects of non-individualized homeopathy may differ across different indications and or different homeopathic products used, including significant and non-significant differences from placebo. So that was our second question. Now, so far, we've looked at methods, results, and quality of data for homeopathy as such. But then we also assessed results and quality of data in comparison to other research fields using the same methods in order to kind of see what does it mean compared to the rest of the research world. Is it very bad? Is it very good? Is it somewhere in between? Now, the most precise comparison was already included in the Shang meta-analysis because the authors had compared their 110 homeopathy trials to 110 trials in conventional or general medicine using identical quality criteria, which is a key requirement, but they also had them matched for similar indications and similar outcome measures. So it's you know, if it's pain in the other one. It's not survival. As you see, the proportion of high quality trials, the green column, was more than twice as high for homeopathy than for conventional medicine, and the difference was significant. So this is the most precise comparison we had. Now, for the two homeopathy meta-analyses by Mathy, 2014 and 2017, with, as you see, 32 and 75 trials, respectively, we found a paper on 1,242 Cochrane reviews and 424 other systematic reviews using all the identical Cochrane risk of bias tools, which was available at the moment, except the authors of this paper, they did not include one item, which was later removed from the risk of bias tool. So we did the same to our two meta-analyses. And as you see, the proportion of high quality trials was, and the distribution of quality showed a similar pattern across all four groups, except you see the non-Cochrane reviews having less trials but higher risk. I don't know if you can see my cursor moving when I do this, but it's the red to the right is smaller and there are more yellows. So there are less trials with higher risks and more with uncertain risk. But these comparisons are not matched for trials and indications. And so they are less exact. It's more sort of order of magnitude comparison. And finally, we compared the quality of evidence for efficacy of homeopathy to a study of 608 Cochrane reviews from roughly the same period, at least the average. And now, as you remember, we had rated quality of evidence as high for individualized homeopathy and moderate for the two other types, non-individualized, and for all types assessed together. Now, for more than half of the Cochrane reviews, quality of evidence was rated as low or very low. So this looks very pleasing for lovers of homeopathy, but this difference should be interpreted with utmost caution because it is not exactly apples and apples. Because there is the thing, most of these Cochrane reviews will have covered one or few interventions for one indication. So some of them will have a small number of trials, and that increases the risk of being downgraded several steps. While our sample had a large, our meta-analysis had, well, from 18 to 110 trials, so it lowers the risk of being downgraded because you have a larger group of studies to support your evidence with. So it shouldn't be over-interpreted, but what we can say is our systematic review dealt with the question if homeopathy does work beyond placebo in general. And for that research question, the quality of evidence was high or moderate, and it was not low and not very low, which is indeed found in clinical research. So how do we interpret these findings and what do they mean? Well, according to this systematic review, homeopathy can have positive effects beyond placebo on disease in humans. And this is in accordance with numerous, I mean, there are hundreds and hundreds of laboratory experiments showing effects partly replicable, can be replicated with the same test system, which is not always the case because laboratory testing is often vulnerable, sort of a little shaky. But the majority of these experiments show effects of homeopathically potentized preparations, including high potencies in physical, chemical, in vitro, plant-based, and animal-based test systems. Still, the skeptic will talk, well, what about the high potencies? Can it work? Well, that is beyond the scope of this presentation. Implications for practice and policy. Our first question was about efficacy of homeopathy beyond placebo. Now, the evidence we found provides no justification for regulatory or political actions against homeopathy in healthcare systems. That's clear. And our second question was, is it meaningful to talk about one common homeopathy effect? For individualized classical homeopathy, that is so. And therefore, it would make sense to have an update of the last meta-analysis on this group of homeopathy by Robert Matthew 10 years ago. That could make sense. But for non-individualized homeopathy, there was statistical heterogeneity, which was, in a way, it was resistant to all, it stayed there, whatever you did to the data. And that means there is not one single effect, and therefore future research should rather focus on specific non-individualized homeopathic interventions or specific indications. To sum up, we found significant positive effects of homeopathy beyond placebo in 5 out of 5 available meta-analyses of all included trials, and in 3 of 4 meta-analyses of high-quality trials. And the homeopathy data had at least similar quality than other correspondent data on other corresponding treatment pertaining to the quality or risk of bias of trials and to the quality of evidence for positive effects. Notably, this does not mean that homeopathy has similar or stronger effects than other treatment, as I read somebody had summarized somewhere. Our question was not magnitude of effects in comparison to other treatment. It was, does it have effect beyond the placebo? So this is a different research question investigated in other types of studies called comparative effectiveness studies. So we cannot say anything about that here. And finally, there was no support for the alternative hypothesis of no outcome difference between homeopathy and placebo, because the data clearly do not show that. Thank you for your attention.